Hi, my name is Courtney Anderson, and I'm an application scientist here at ACD. I'm here to present a study that we recently conducted in our labs entitled The Detection of Immune Cell Checkpoint and Functional Markers in the Tumor Microenvironment by the RNA in situ Hybridization RNAscope Assay. And I'd first like to start just by giving a brief background on cancer cell immunity. The generation of immunity to cancer is a cyclic process that can be self-propagating. It starts by the release of cancer cell antigens from the tumor cell. These cancer antigens then get presented on either dendritic cells or antigen-presenting cells, also known as APCs. There's priming and activation of the APCs and T cells, and then trafficking and infiltration of the T cells into tumors. The tumor cells are then recognized by the T cells, and then killing of the cancer cells then occurs. And this cyclic process then starts all over again. Now, this can lead to an accumulation of both immune stimulatory factors, which help to amplify and broaden the T cell response, as well as immune inhibitory factors that halt the development of or limit immunity. Now, tumor cells have taken advantage of this and can actually hijack this system in order to evade the immune response. When tumor cells are evading the immune system, the tumor cells bind to T cells and can actually deactivate them or turn them off. Immunotherapy takes advantage of this process by actually blocking the tumor cell from deactivating the T cell. And it does that by either targeting inhibitory or stimulatory factors that are involved in the cancer immunity cycle. And immunotherapy drugs can be in the form of either vaccines, monoclonal antibodies, checkpoint inhibitors, cytokines, and chimeric antigen receptor or CAR therapy. And stimulatory and inhibitory factors of the cancer immunity cycle can include immune cell markers, immune checkpoint markers, and also cytokines, chemokines, and their receptors. And several of these molecules are actually current targets of immunotherapy, such as PD-1, PD-L1, and CTLA-4. In this study, we examined 3 categories of markers. We looked at 12 checkpoint markers, 14 immune cell markers, and 24 cytokine and chemokines, as well as some of their receptors, totaling 50 markers analyzed using RNAscope. And we're currently testing more markers in the R&D lab, so you should stay tuned. The first experiments that I'd like to demonstrate the immune cell infiltration into the lung tumor. I show here lung cancer and ovarian cancer. I'd like to focus on the lung cancer first. In the lung cancer, we examined the expression of PD-L1 using green chromogen and CD8 alpha using the red chromogen. And what we're able to see is a high abundance of CD8 positive cells that are actually both in the tumor and the stroma. And many of these cells co-localize with PD-L1 positive cells. So what this is suggesting is that most likely the CD8 positive T cells are inducing PD-L1 expression in the tumor cell. Now, on the flip side, in the ovarian cancer sample, we still have CD8 positive expression in some cells, both in the tumor and the stroma. And there's much fewer PD-L1 expressing cells. So most likely these PD-L1 expressing cells are immune cells and not actually tumor cells. Another experiment that we wanted to perform was to look at T cell infiltration into the tumor. And so again, using ovarian cancer and lung cancer, we performed an assay using CD3, CD8 alpha, or CD4 in green chromogen. And we did this in combination with interferon gamma using the red chromogen. And what you can see in both tumor samples is that we have expression of all the CD markers. And I should mention that CD3 is a marker of all T cells, whereas CD4 and CD8 are markers of subsets of T cells, and they are mutually exclusive, so no 2 cells will express CD4 and CD8. So what we see is co-localization of the CD markers with interferon gamma in both the ovarian cancer and the lung cancer. So this data demonstrates that we are getting T cell infiltration in the tumor. And then we wanted to see if we could specifically identify regulatory T cells in the tumor and the stroma. So again, using ovarian and lung cancer samples, we performed an experiment using CD4 detected by green chromogen and FOXP3 detected by red chromogen. Now, FOXP3 is a transcription factor that specifically marks regulatory T cells. And what we see in both of these cancer samples is that while we have a high number of cells that express CD4, we have several cells that express both CD4 and FOXP3. So this demonstrates that we are actually detecting regulatory T cells in both the tumor and the stroma. Now, what I show here on the screen are a panel of immune checkpoint markers that we analyzed in our study. I show here 8. We looked at 12 checkpoint markers in this study, and I'd just like to highlight a few of these. We have IDO1 in the esophagus, which is a very high expressing cell. We also have PD1 and PDL1, which are much lower expressing immune checkpoint markers. We looked at 14 immune cell markers, and I'm showing 8 of them here. Again, just to highlight a few of these, as I mentioned before, CD8 alpha we've shown in the duplex assays. We also have high expression of HVEM, and then we can also detect expression of some of the lower markers, including ICAM1 and OX40L. And then lastly, we looked at 24 cytokines, chemokines, and their receptors. And some of the most highly expressing, such as TGF-beta1 in the stomach, is quite robust, but we can also detect expression of some lower expressing markers, including CX3CR1 in the liver. Now, there are 2 markers that were particularly challenging, and they're both cytokines. The first one I'd like to point out is interferon gamma. And we found this to be a very challenging target to identify because it was very hard to find tissue that expresses interferon gamma at abundant levels. It could be the biology, that this marker is just a very low expressing marker in the tumor cell, but we are working on trying to identify tissues that have more robust expression of this marker. We'd just like to point this out because customers may have a hard time detecting this in their tissue samples as well. And it may not accurately represent the quantitative aspects of expression. Now, another cytokine that was also problematic was TNF-alpha. I show an example here in the breast tissue where we can see more expression than interferon gamma, but it was still challenging to find a tissue that had high expression of TNF-alpha. Now, interleukins, we've had much better success with this class of molecules, as well as chemokine detection. It could be the biology where both of these markers are more highly expressed, but we are performing experiments to get more data to support this hypothesis. So in conclusion, I'd just like to summarize that the RNAscope manual Brown assay was able to detect 50 immune checkpoint and functional markers in multiple human tumors. We detected 12 checkpoint markers, 14 immune cell markers, and 24 cytokines, chemokines, and their receptors. I showed a subset of those images in this presentation, but you can find all the images in our appendix at the website listed on the screen. The manual RNAscope duplex custom assay was used to demonstrate the localization of PDL1 and several immune markers in both human ovarian and lung cancer samples. And so overall, these results demonstrate the utility of the RNAscope technology in studying tumor immunology and key targets of immunotherapy in both the tumor and its complex microenvironment. Thank you.